All right, let's just jump right in. I want you to forget everything you think you know about the cell membrane being just some simple wall. Instead, I want you to picture this: a bustling, sprawling metropolis we're going to call Cell City. And its border? It's not just a wall. It's an intelligent, dynamic, and incredibly busy border control station. This is where all the action happens. And the number one golden rule at this border, it's something called selective permeability. This is the core mission, to clearly define what's inside the city versus what's outside. It's about meticulously regulating every single thing that tries to cross. It's like the world's smartest bouncer, letting all the good stuff in, keeping the bad stuff out, and just maintaining perfect order and balance inside the cell. So, here's our itinerary for this tour of Cell City's border. We're going to cover passive transport, that's the easy stuff. Then, active transport, when things get a little tougher. After that, bulk transport for the heavy lifting. And we'll finish it all off with a quick recap of our journey. First up, let's talk about the easiest way to get into Cell City. This is passive transport. It's basically for molecules that are just, going with the flow. They're moving from a really crowded area to a place that's less crowded. And the best part? No energy pass required. It is completely 100% free. Now, this free travel comes in two main flavors. On one hand, you've got simple diffusion. This is for the real VIPs. I'm talking small, nonpolar molecules like oxygen and carbon dioxide. They can just slip right through the membrane's fatty layer. No questions asked, no help needed. But on the other hand, you have facilitated diffusion. This is for molecules that are a bit bigger or have an electrical charge. They can't just sneak by. They need a little help from a protein escort to get across. And that help they get? Well, it comes in two forms, and you can think of them as express lanes at the border. As you can see here, channel proteins are basically like pre-made tunnels that are specific for certain things, like ions or water, to just pour right through. Carrier proteins, though, they're more like a personal escort service. They physically grab onto a specific molecule, like glucose, and carry it across to the other side. And get this, these channels are incredibly smart. They aren't just open all the time. Oh no. Take these voltage-gated calcium channels. They act like these high-tech security gates that are totally sealed until they detect a very specific change in the electrical charge across the membrane. And when that charge shifts just right, bam! The gates swing open, calcium ions flood in, and that triggers everything from muscle contractions to neurons firing. It's amazing. Okay, so if channel proteins are tunnels, then carrier proteins are like a revolving door. Let's look at the glucose escort service, this protein called GLUT4. It starts with its outer gate open, just waiting. When a glucose molecule comes along and binds to it, it's like a key in a lock. That binding triggers a shape change. The outer gate snaps shut, and the inner gate swings open, dropping the glucose safely inside Cell City. And all of this still requires zero energy from the cell. Of course, we can't forget about water. The movement of water is so important it gets its own special name, osmosis. Water has its own dedicated VIP channels called aquaporins, and it's always moving to try and balance things out. It flows from an area with less stuff dissolved in it, what we called hypotonic, to an area with more stuff dissolved in it, or hypertonic. Okay, that's all fine when you're going with the flow. But what happens when Cell City needs to import something that's already in really short supply outside? Or what if it needs to export waste that, frankly, doesn't want to leave? Well, you can't just go with the flow anymore. You have to actively fight against it. And that comes at a price. I mean, this is the crucial question, right? How in the world does a cell force molecules to move from a place where there are very few of them to a place where it's already packed? It's like trying to swim upstream or push your way onto a subway car that's already overflowing. It's just not going to happen on its own. The solution, my friends, is active transport, and this is a process that needs power. Specifically, it uses the official energy currency of the cell, a molecule called ATP. Just think of ATP as the toll you have to pay. It's the cash you need to get through the special powered gates at the border. Now, there are basically two ways to pay this toll. You've got primary active transport, which pays directly. It burns an ATP molecule right there on the spot to power a pump. Then you have secondary active transport, which is a bit more clever. It doesn't use ATP itself. Instead, it takes advantage of the energy that was already spent by a primary pump. It's like one molecule hitching a ride with another that's flowing back down the hill that the first pump created. So let's look at the main pump that sets all this up, the sodium-potassium pump. I mean, this thing is an absolute workhorse. It is fundamental to almost all animal life. For every single molecule of ATP it burns, it forcefully pumps 3 sodium ions out of the cell and pulls 2 potassium ions in. And because it's moving things in opposite directions, we call it an antiport. So why does it do all this work? Well, all of this hard work pays off big time. By constantly pumping these ions, the cell builds up a really steep electrochemical gradient. That's a huge difference in both chemical concentration and electrical charge. This gradient is essentially a battery. It's charged up and storing a ton of potential energy that the cell can tap into later to power other critical tasks. And here is where we cash in on that battery. This is brilliant. The SGLT1 transporter opens its gate. Now remember, all that sodium that was just pumped out, it is desperate to get back in. So it rushes down its gradient with a lot of force. The transporter cleverly uses that rushing force to drag a glucose molecule right along with it, even if that means pulling the glucose against its own gradient. And since they move in the same direction, it's called a symport. Okay, last stop. What about the really big stuff? How does Cell City import an entire protein or export a massive batch of hormones? For that, we need a completely different system. It's time to call in the Special Delivery Service. Welcome to Cell City's main loading dock. To bring big cargo in, the membrane itself actually folds inward, wrapping around the material and basically swallowing it whole. That process is called endocytosis. To ship big cargo out, a little bubble-like package called a vesicle travels from inside the cell to the edge, merges with the membrane, and releases its contents to the outside world. And that's exocytosis. Whew! All right, that was a whirlwind tour of the border, wasn't it? Let's do a quick final review, just to make sure all our travel documents are in order before we leave. This little cheat sheet really sums it all up beautifully. You can see the 2 types of passive transport, diffusion and facilitated diffusion, neither of which require any energy. Then you have active transport, which always requires energy, either directly or indirectly, to fight against the gradient. And finally, you have bulk transport for the really big jobs. Each one has a very specific mechanism for a very specific purpose. So I really hope you'll agree by now that the cell membrane is so much more than just a static wall. It's a living, breathing, thinking gatekeeper. It's constantly making decisions, spending energy, and running all the complex logistics needed to keep the entire city within alive and thriving. And here's a final thought I want to leave you with. Every single nutrient you absorb from your food, every nerve impulse that lets you think and move, every single medicine that works inside your body, all of it relies on these tiny molecular gates functioning perfectly. It really makes you wonder, doesn't it? What are the consequences for our health when just one of them breaks down?